So I'm going to be pretty superficial, but happy to unpack. I have 3 stories I'm very excited about, especially the last story, which you haven't heard before. Can you see this? Okay. Everybody good? So vignette 1 is biochemical annotation of all possible missense variants of a protein of interest to find those that cause misfolding and degradation. This we called voodoo last year, and we're going to talk about MLD. So 6 or 7 years ago, Xinying Hong in my lab developed newborn screening for MLD. And then you heard the German study in France and the MLD Alliance. So we've screened about 600,000 babies now prospectively and live. And there's no false positives, really zero. Sorry, nobody believes me. 0 is 0. 0 is a funny number, except when it's 0, right? Anyways, and as far as false negatives, we had 15 blood spots newborn, mostly California, that from kids diagnosed later with MLD. We got their newborn, and they all tested positive with the sulfatides. And then England got another 25. So 40 out of 40 newborn blood spots, we don't miss them. So we think the false negative rate is essentially 0. And there's no better way to prove it than that. So if your sulfatides are high and your ARSA is low, you are going to get MLD. It's a question of when. You're going to get MLD. Can we predict the age of onset? And this is going to be mostly the genotype, as Laura mentioned. So 3 years ago, we measured the ARSA enzymatic activity when we express it in human cells for about 250 missense variants. But more recently, we've put all possible missense variants, means every amino acid change to all 19 other options, so 10,000 variants in human cells, and we measure whether the protein is degraded. Is it unstable and degraded? Most genetic disease is caused by missense mutation that causes protein instability. Not always, but that's the major cause. It's kind of like Survivor Island. A bunch of people board a boat, and we put a name tag or an ID bracelet on all the people. They go on the island, they duke it out, and then they go home, and we read all the bracelets and see who did not make it. That's exactly what we do. The way we do this is kind of clever. So this is an ARSA variant, S395T. And every cell expresses a single variant. And it has a little blue protein called the MCP. Comes from a bacteriophage. And it binds a circular single-stranded RNA because the RNA has a hairpin that binds tightly to the MCP. It's a circular RNA. It cannot be degraded because RNA is degraded from the ends. But it has a little sequence, the barcode, the name tag. Imagine trying to measure the abundance of 10,000 different proteins at the proteomic level. It's impossible. But imagine trying to measure the abundance of 10,000 name tags at the next generation sequencing level. It's trivial. So we read the name tags, and the name tags tell us the survivors. This works. For example, this is a Western blot would be the conventional way to do it. You take the cells, you run it out and see how much protein's left. So the wild type, you can see the glycosylated, non-glycosylated forms. The half-life of wild type ARSA is a few days. You see it starting to go down. These are 2 pathogenic variants, G110, 101V and R246C, that are mostly gone at 48 hours. And that's exactly what we see in the voodoo data, okay? So, okay, anyways, we have this data for all 10,000 possible mutations. I'll give it to you now. Now, this is interesting. So we take all the missense variants in ClinVar for ARSA. Now we say late infantile. What does that mean? Because it means the variant is found in a late infantile patient, and every late infantile patient has 2 severe variants, okay? So any variant you found in a late infantile is severe. And then we have patients that have a severe, and then another one that's not severe that we find in juvenile. So we say that juvenile variant is a juvenile variant because when paired with a severe, it's juvenile, et cetera. So now we have the stability scores for all of the variants in ClinVar, and you can see that the late infantile has the lowest, juvenile. They stack nicely all the way up to wild type, but there's a few exceptions. There's a few troublemakers. There's a patch of juvenile variants that have high stability. And we know these are on the surface of the protein that are involved in the dimer-dimer interface and in the active site. So if I tell you the stability of the protein is okay, you cannot say it's benign. If I tell you it's below 58%, I guarantee you it's severe and it contributes to LI and LD. So I'll come back to this problem that we cannot say it's benign, we can only say it's bad. Now here's the real test. And this is really remarkable. So we took a DANGS 90 MLD patient cohort, all diagnosed with MLD. We know the age of onset, we have the genotypes. We applied the easy peasy European consensus guidelines for going from genotype to phenotype. 27 authors on the paper. Consensus for these principles reached by 90% or more people. It's very simple. Any newborn with LI, those variants are severe. Any two severes in any combination is LI. Everybody in the MLD field thinks that's true. Family history, one result transfers to the next member. That seems reasonable. There are two late onset mutations that are well understood and very common. They drive early juvenile or later onset MLD. These are the European consensus rules. So how well do they work? 89 patients from Laura. There were 4 that she says have MLD, but we cannot find the genetic basis. One of the alleles has ClinVar benign and in vitro benign variants. So we think the genotype is missing a variant. So we exclude these 4 patients. We don't understand these genotypes. That leaves 89. Of 85, 65, we can apply the easy peasy European consensus. We get the age of onset correct 100% of the time. Now we can try our protein stability survivor thing on 56 of the 65 because they have at least one missense, right? We do missense. We get it right 98% of the time, 55 out of 56. What does right mean? It means we say early. Early means late infantile, early juvenile. Qualifies for gene therapy. What does late mean? Late juvenile adult does not qualify for gene therapy. So if we say early, means it's LI or EJ. If we say late, it's late juvenile or adult. Now we take the 20 out of 85 where we cannot apply the European consensus guidelines. And of these, we get it right 19 out of 20 times, 95% of the time. So we can predict prognostically at birth the age of onset of MLD in almost all cases. This is a perfect, essentially perfect newborn screening. I can show you the data. Then yet two. We have a problem. If we say the stability is high, we cannot say it's benign. So if you have a kid with high sulfatides and low ARSA, you are going to get MLD. It's a question of when. If the stability, if we can't apply the European consensus and we rely on stability and one of the variants comes out folded, we cannot say set the baby free. It could be a bad guy. This is a rare case, but what we need is fast, on the fly, CLIA lab, easy enzyme activity. Because every time these cases where the protein's stable but it causes disease, the enzyme activity is essentially zero. So if we get a new variant in third-tier newborn screening, we need on-the-fly unpacking enzyme activity if the stability data says it's okay. Let me show you how we're going to do this. It's really cool. $200. You call up Twist Biosciences. They send you the single ARSA variant. Within a week, it comes in a plasmid ready to go. The CLIA lab thaws a tube of engineered cells, adds the DNA, waits 2 weeks. Lyse the cell, measure the ARSA activity by mass spec. A little bit of liquid transfer, super easy. Now this gets you expression of ARSA at the native level. It's not overexpressed. There's people that are worried about overexpression in these in vitro systems. I don't think it's a problem, but anyways, it's native expressed. It comes from a single site in the chromosome, so we don't have to worry about epigenetic funny stuff. And it's measuring only the enzyme activity in lysosomes for mutations that cause mistrafficking. Now I want to talk about my favorite story, cystinosis. What about diseases like this one, supertreatable lysosomal storage disease? There's no possible biochemical assay on the horizon. It's a transporter that pumps cystine out of lysosomes. There's no way to do a functional assay of a transporter in a blood spot. The DNA people say the only way to screen for cystine is very treatable disease. Only way is DNA, but they're wrong. I'll show you a biochemical way to do it. It amounts to measuring the abundance of the protein in the blood spot. Now, wait a minute. Sometimes you're going to have mutations that cause cystinosis and the abundance of the protein is normal, and you're going to have a false negative. But I tell you, that's the false negative, that will be much, much less than if you did it by DNA first. Most of the time, we think more than 90% of the patients will have small amounts of the transporter in the blood. But how do we measure it? So David Baker won the Nobel Prize in chemistry 6 months ago. He's been my colleague and friend for 40 years. What he has done is absolutely remarkable. And I got him to play with me in the newborn screening world, along with Kezia and Yushin, and Sihun, my longtime collaborator, Silchins. So the idea is we're going to take the blood spot, we're going to trypsinize it to generate a bunch of tryptic peptides. One short peptide will be the signature peptide. If we quantify that by mass spec, it tells you how much cystinosin transporter protein you have. The peptide is unique. We can design in 2 days a protein from scratch that binds to any given peptide, 100% selective with very high affinity. So what we can do is we can add this protein to the blood spot extract after we trypsinize and pull out this peptide. You see, if we trypsinize all the proteins in blood, there's billions and billions of peptides. We'll never find the needle in the haystack. We need enrichment. Now, we can design a protein that is a perfect three-dimensional jigsaw puzzle for any peptide of any sequence you want in 2 days. So we take a 3 millimeter blood spot, we digest with trypsin for 2 hours. We add beads containing our capture protein, we elute the peptides and do mass spec. Let me show you that it works. This result is a month old. So what you have here is on the bottom here, you have a normal blood spot. You see a nice peak for the cystinosin-derived peptide, and here's 1, 2, 3, 4, 5 cystinosin patients. The peak's gone. How do we know this is cystinosin? Well, we don't know for sure, right? Only, somebody knows, but this is science. He doesn't tell us this is cystinosin. So it has the right retention time. It has the right fragment mass for 5 fragments in the right ratio. And best of all, it's missing in cystinosin patients. I think that is the cystinosin peptide. Now we can screen for cystinosis biochemically and very easily. And we can do this in less than 1 minute per baby, making it the world's fastest newborn screening assay ever developed. Now we go on with Sihun. We can multiplex this with hundreds and hundreds of diseases as long as the protein reveals itself in blood. So it has to be a protein in the blood cell. If it's a brain-specific protein, we would need a brain biopsy, right? But there's plenty of diseases in blood we can measure the protein. So right now we can do Wilson disease, which is a copper transporter, super treatable. Cystinosis, all of the primary immunodeficiencies that Joanne can treat. Wiskott-Aldrich syndrome, DOCK8 deficiency, A-gamma, whatever it's called. So easily we can do hundreds of diseases at the same time and design binding proteins for all of these things, I would say inside of a year. In a few days, we can get a protein that binds any peptide you want from any protein. Okay. And that's it. Thanks.